In this lecture, we'll discuss about gene cloning or DNA cloning. Gene cloning or DNA cloning refers to the production of large number of identical DNA molecules from a single DNA molecule. Usually, the gene that we're trying to clone has some significance, like the human insulin gene, which is used in the treatment of diabetes, or the Bt gene obtained from the bacteria Bacillus thuringiensis, which has its application in the production of biopesticides. There are two approaches used in gene cloning: the cell-free approach or the cell-based approach. The cell-free or in vitro approach is nothing but PCR, that is polymerase chain reaction. As we know, in PCR, we don't use living host cells. We rather simply add the DNA template, primers, and DNA polymerase in a test tube and amplify it in a thermocycler machine. Whereas in the cell-based approach, we use living host cells to replicate and form multiple copies of our target gene. The method used is referred to as the cut-paste method. That is because we cut a gene of interest and paste it with the vector and then insert it into the host cells to form multiple copies. The first step in gene cloning would be to obtain cells containing the desired gene. That is, if I want to clone the Bt gene, I will first culture Bacillus thuringiensis. Once I have my cells, the second step would be to isolate DNA from the cells. Once I have my DNA, I need to cut out my target gene from the DNA. We don't want to clone the whole DNA, but merely only a part of it, that is our target gene. In order to achieve this, we use special enzymes known as restriction endonucleases. These enzymes cut the DNA at a specific site. There are many types of restriction endonucleases, and we'll choose a restriction endonuclease that has a site specific for the sequence that is present in our target gene. We use the same restriction endonuclease to cut the vector DNA molecule. This vector is a carrier that transports our gene of interest into the host cell. These restriction endonucleases cut the gene unevenly, due to which the ends do not have base pairs. The same thing happens with the vector molecule. So once we paste them, or that is attach the vector with the gene of interest, they form complementary base pairs. This attachment or pasting of the vector with the gene of interest is done by the help of the enzyme ligase. Now, as the vector molecule has a foreign DNA attached to it, it is now known as recombinant DNA molecule or rDNA molecule. Hence the name rDNA technology. Once the recombinant DNA molecule is ready, we insert it into living host cells. We can use prokaryotic cells or eukaryotic cells. This insertion of recombinant DNA molecule inside the host cells is known as transformation. Once transformation is successful and we have the rDNA molecule inside the host cell, we have to culture the host cells and then we can obtain our gene of interest from it. So this is an overview of gene cloning, and we will discuss each step in detail in subsequent lectures.